Okay, well, everyone, welcome to this week's Work in Progress series. This week we have Dr. Megan Linske, who is a researcher at the Connecticut Ag Station with a background in wildlife biology and ecology. Her expertise allows her to approach tick management from an ecological perspective, ensuring that strategies are not only effective, but also sustainable. Dr. Linske specializes in developing integrated pest management techniques that balance the need for controlling black-legged ticks with the protection of non-target species, such as pollinators. Her work emphasizes the importance of understanding ecosystem dynamics to create environmentally responsible solutions for tick control. Megan, thanks for joining us.
Thank you for having me. I'm very excited to talk to everybody today. And hopefully I see a couple of familiar faces and names on my other screen over here. So I apologize if I'm sounding redundant with things you maybe heard before, but hopefully some new stuff I can share with everybody about tick control, specifically looking at sustainable spraying and its impacts on non-target species. So I should also preface this by saying this is a combination of studies that I'm going to be talking about that I've been doing with Dr. Scott Williams. I'm sure most of you guys are aware of him and his research. And so this has been done in that collaboration. So just a little background and context for those of you, I'm sure most of you guys are familiar, but I'll still cover kind of the basics for those of you who are not. There are between 10 to 16 different species of ticks throughout the Northeast, depending on what state you're in. But in the state of Connecticut, there are 5 that we consider of medical importance that we focus on. The most obvious one is the black-legged tick or deer tick, the American dog tick, Lone Star tick, Gulf Coast tick, which is relatively new and very sparse, but still present, and the Asian longhorn tick. And the reason for these specific species is they're all human biting. While not all of them transmit pathogens, they all have been recorded to bite humans in the state of Connecticut. So that's what we're keeping an eye on them. Black-legged tick is obviously the most prevalent one. American dog ticks are probably secondary depending on season. And then we have the couple that are relatively new in establishing in the state of Connecticut and in varying densities. Lone Star and Gulf Coast tick being more southeastern species, and the Asian longhorn tick, actually I think the name's changed to longhorn tick, which is an exotic introduced species. That being said, there is a reason why a lot of our research is focused on black-legged ticks. So through both our passive and active tick surveillance program, the predominant species that are submitted or collected are black-legged ticks. As you can see here, out of almost 6,000 identified individuals, more than 4,000 of them were black-legged ticks. And then, like I said, second was dog ticks, and then a smattering of Lone Star ticks and other species such as Haemaphysalis longicornis. And the same can be said for our active tick surveillance program, which is a program that spans 40 different sites throughout the state of Connecticut, covering all 8 counties. And same thing, very similar results where we collected over 5,000 ticks. This was from 2023. Almost 4,000 of them were the black-legged ticks, and then kind of distributed amongst those other species. And then in addition, not only are they the highest proportion of ticks that we collect, but they're also one of the species that continues to kind of increase in its density and distribution throughout not only Connecticut, but the rest of the Northeast, and kind of moving out west a little bit as well. There's a reason why they're important and a focal point for a lot of our studies. In addition, in the state of Connecticut, the nymphal stage is usually about 20 to 30% infected with Borrelia burgdorferi, the causative agent of Lyme disease, and the adults, like this female seen here, are upwards of 60%. So from the medical and public health perspective too, that's why they're a major focus point for us, and why we're encouraged to do these studies looking at sustainable and long-term management strategies. While the focus is always on black-legged ticks, when we quantify the efficacy of our treatments, we're usually surveying that particular species. All of these are species that will be impacted by our treatments. They'll vary a little bit in behavior, especially when we look at things like Lone Star tick and longhorn tick that are a little bit more predatory and faster moving. Some of the applications might be a little less impactful, but they'll still have some impact in reducing those tick species as well. Which brings me to our next point. So the focal point is black-legged ticks, as well as any other tick species that happen across the landscape in Connecticut. And from our perspective here at the Ag Station with our program and tick team, we are focused on something called integrated tick management. So while we do have our passive and active tick surveillance programs, which kind of monitor and survey the landscape, we then take that information and then we use it to appropriately optimize management strategies based on tick species that are present and in what densities, and then also what's the greatest risk to public health. So this means what we're doing is we're taking individual approaches, optimizing them based on the information that we have, and then using them in an integrated approach. And so individual methods for ITM span a whole gamut. We don't do all of them here directly. So like education and behavior changes, as well as personal protective measures, are things that are kind of outside our wheelhouse, but we indirectly have impacts on. We have done work on landscape modifications, both chemical and biological control, which we'll be talking about a little bit today. We've done things like host reduction and exclusion, which can be a lot of time and effort, and not necessarily the greatest outcomes and quite contentious at times. But we've also looked at host targeted acaricides and vaccines, and I'm sure you guys have seen between a couple of our team members some of those talks about recent advances in those areas. But today I'm going to be focusing on things like chemical control and how we've altered kind of the application of those things to be more effective with less environmental implications. So the first part of this is a study that we did a couple of years ago looking at inventorying people's backyards. So this was done in conjunction with funding from CDC. Basically, CDC has determined that, along with other research as well, that the greatest risk to people, the greatest risk to them encountering ticks is in their paradomestic habitat or the habitat surrounding their backyard. There's a bunch of reasons for this. Some of them are the fact that that ecotonal area is prime habitat not only for ticks to be out questing and possibly encountering hosts, but just also like host species like white-tailed deer, white-footed mice tend to prioritize those areas as well for a multitude of reasons. The other thing too is when people are in their backyards, they might not be as vigilant or their behavior might change due to a level of comfort. And so they might be encountering ticks at greater encounter risk there, but they might not be checking as often. Or likewise, they might have outdoor pets that come in, or children for that matter, who utilize that same area and then bring in ticks. So we were asked to kind of go back into those backyards and see which areas were possibly correlated with higher tick densities and therefore a higher risk of tick encounters for homeowners and kids and dogs and all that good stuff. And so we had all these different variables that we looked at, and then a couple of manipulations as well. So not only were we looking at habitat structures, things like vegetable gardens, ornamental plantings, hardscapes, terrascapes, etc., but we were also looking at the presence and absence of things like wood piles, stone walls, bird feeders, and then even things like a wood chip barrier or leaf litter removal, things like that. And this also hinges on a study that was done back in 1993 by Dr. Stafford and Magnarelli that determined that the greatest risk of encounter for ticks, like I said, is in that ecotonal zone, but within the first 0 to 3 meters in, with a focus really on that first meter, is almost 70% of the ticks that you're going to encounter. This was great information, and I think this really not only spread awareness to the public and homeowners, but also kind of guided a lot of pesticide applicators when they went into their backyards and they knew where to kind of prioritize their spray. However, this still led to broadcast application to the entire area. And as we know, if you go out, walk into your backyard, it's not this nice uniform strip of habitat that's exactly the same throughout. There's, like I said, there's hardscapes and xeriscapes, there's low shrub layers, leaf litter, stone walls, wood piles. There's all sorts of different structures in place there, right? And so we wanted to quantify those areas to see maybe if there's some parts that should be prioritized for higher tick risk versus areas that were probably a little safer or lower density of tick species. And then in addition, putting in some of those modifications like a wood chip barrier, which has been kind of traditionally used in the past, as well as some leaf litter removal activities too. And what came out of that study was interesting, but also expected. So if nothing else, it kind of gave us descriptors for the areas that had higher tick densities versus just like I said, that broadcast application or that broad scope of saying your entire ecotonal zone is a high tick density area. So things like center portion of the wooded area with leaf litter, wooded area with leaf litter along short grass ecotone, center portion of wooded and brushy area, and the short grass lawn along the wooded ecotone. So those are all descriptors of that area, like I said, from that lawn edge working into the woods about a meter or so. That's the classic leaf litter, shrubbery understory, invasive kind of area that we'd expect ticks to be in, as well as a lot of their host species. It provides ample cover, as well as questing area, and then that nice cushy leaf litter area for them to retreat after questing when humidity levels are not ideal. And then what was interesting about this stone walls versus wood piles, we speculated that maybe they'd be the same because they're similar in structure, but it makes sense because wood piles are in a constant state of rearrangement, right? So they're either decaying, they're being moved, they're being added to, relocated, used. So it's not very constant versus stone walls, especially in much of the Northeast, in particular Connecticut, are historic structures. So actually, I think it's legal to move or alter them without permission. So they are what we call like mouse highways and hotels. They are pretty constant structures. So it makes sense that they're this fixture that provides not only like a pathway, if you will, but also housing and shelter for key species like white-footed mice, chipmunks, that sort of thing. And then interestingly enough, the wood chip barrier didn't really have an impact, which makes sense when you think about it. I mean, especially with black-legged ticks, most of their movement is 2 to 4 inches vertically on vegetation substrate. So they're not really traversing over a wood chip barrier, whether it's there or not. They're not going to be crossing into a nice habitable area into a short grass area because it's not, that wouldn't work for them. Any transfer of those tick species into the lawn area, it's most likely due to being distributed by a host species. Like a deer is not going to care if there's a 3-foot wood chip barrier. They're just going to go right over it. We think, if nothing else, the wood chip barrier is more of a visual reminder for homeowners to not enter into the tickier habitat, but not necessarily something that negates the distribution pattern of the tick itself, if that makes sense. And then as for the leaf blown area, these were just small parcels that we did in people's backyards. So we speculated that it might not have been enough of a area to edge impact on the ticks to really see a difference. Granted, if you wanted to fully leaf blow your entire backyard, that might be a different scenario, but we did not do that in people's backyards. Like I said, this was interesting because it provided us with specific details on which areas were in fact tickier or higher density tick areas versus others and why, and it makes logistical sense. However, this just further supported that with quantitative data. This also kind of further solidifies the fact that this is not just where ticks are and where the habitat is as well, but also the host dynamics that are at play. And like I said, this is kind of a key intersection area between key host species, habitat structure that fits the tick demographic, the ticks themselves, and then also where people and their pets tend to spend a lot of time as well. So the hope is with this is not only to be like, okay, yeah, now we know where all these higher tick density areas are, but maybe we can also, in response to that information, have more targeted approaches to a Acaricide application. So instead of going out to a backyard and being like, okay, the 1 meter zone surrounding this person's 1 acre lot, and spraying the whole thing, regardless of what the structure is in place. Maybe instead we can kind of have focal areas that we spend our time and maybe walk into someone's backyard, do a proper assessment of what's there, and then prioritize those tickier areas. Likewise, it's also a great opportunity to make homeowners aware. So maybe in that leaf blown area, their risk isn't very high, but if they're walking and dumping their leaf litter into a stone wall adjacent area, maybe they should be paying closer attention or doing a tick check afterwards to negate some of that encounter risk that they might be stumbling upon. We were able to get out this information to a whole suite of different audience members, which was really exciting. Not only were homeowners really excited to see the information, and like I said, it kind of increased their awareness, but also we had a lot of pesticide applicator feedback saying that this was really interesting, and they're hoping to kind of incorporate it into their practice, which is really exciting. And if you want to learn a little bit more about this particular study, it is published. And this is a quick QR code. Hopefully it's still working. It was working when I checked this morning. Sometimes they fall by the wayside. But if you have any questions about that in particular, I'd be happy to answer them after this talk. Which leads me to the next segue part of this. So all that is well and good and great information. Now that we've learned where the ticks are and in what densities, now we need to learn how to properly treat them. And as all of you are probably well aware, ticks are not new to the landscape. I think this reminds me of Jurassic Park, where they took that piece of amber and they pulled the dinosaur DNA out and they like created dinosaurs. This is very similar. So we have the amber, we have the ticks. So it's very clear that they have been here a lot longer than probably the rest of us. So we know that they're hardy, we know that they're resilient, and we know that they're kind of a fixture on the landscape. That being said, they're at much higher densities than they should be, and that's for a multitude of reasons. There's also a much higher pathogen prevalence than we'd like to see. So we need to come up with strategies that better treat them on the landscape and reduce them, and then subsequently reduce the risk to public health. Like I said, they've been around for a while, they are hard to kill. And unfortunately, something that not unfortunately, but something we kind of keep coming back to is we tried a bunch of different strategies for treating for ticks, and they all have variable success, But if we keep coming back to synthetic pyrethroids, they really are just the most effective strategy. That being said, the reason why they sometimes aren't selected is because they are non-selective. They're not specifically geared towards tick species. They're a general anti or a pesticide, so they're gonna attack anything that's on the landscape, including some of our species that we do not want to target. And botanicals and biopesticides have also been used. Variable success, just depending on the type that it is. And it's not to say that they also don't have impacts on non-target species as well. So we're back to researching synthetic specifically pyrethroids and seeing how maybe we can alter the timing a little bit so that we're not necessarily being more selective. We can't make it more selective as a chemical, but we can make it more selective in its timing and application. And so, like I said, the key piece here is the timing that was traditionally used impacts a lot of non-target species. And regardless of whether it's a synthetic or a natural or botanical insecticide or pesticide, it will have impacts to other non-target species. And this is specifically due to the fact that there's a timing with stages and when they're out and questing. And so obviously you are all familiar with the fact that black-legged ticks, which are the focal point of a lot of these studies, and this one in particular, have a 2-year life cycle. Typically the nymphs start coming out in like late spring, early summer, and that's what they're the highest epidemiological risk for the public because they are, like I said, in the state of Connecticut, they have an average infection of about 20 to 30% with burley burgdorferi, but it's also their size. They are so small that they're really difficult for people to find on themselves. Even if you do a tick check, like I always say, if they make it to my hair, they win. Like I'm not going to find it. But so this timing is really critical for when we treat that particular stage. That being said, it's the same time when a lot of other beneficial species are also out and active. We're looking at the three, well, outside of eggs, 3 stages, the ones that move around, I should say. The nymphs tend to be out in that late spring, early summer, and then they kind of go by the wayside. They're still present, they're just overwintering in that leaf litter area. Same thing, that they're still present in the ecotonal area, but they tend to start their quiescing patterns in that like fall time, and adults start to become more active during that same time. That being said, when we look at things like non-target beneficial species, monarchs tend to be the poster child because they are very pretty, and they tend to come in full force during specific seasons, which happen to coincide with spraying season. So you can see by this chart kind of where their migration patterns happen, the timing of those migration patterns, their breeding areas and breeding seasons. And like I said, they all coincide with the same time that these nymphs are out active and questing, and the same time that we're spraying their backyards. So not great. And this is just one focal species that we have information about their migratory and movement patterns and activity patterns, but there are a lot of other ones. Bees tend to be a focal point as well for very obvious reasons, as well as a whole suite of other pollinators, things like beetles. We want to negate some of those impacts. One thing we were looking at in particular was altering the seasonality of sprays. So traditionally these sprays, like I said, they happen ideally like the beginning of May ahead of the nymphal peak. This is for the state of Connecticut. Obviously it changes depending on where you are in the Northeast. But this is when it typically happens. And this has been traditionally done. They do that broadcast application throughout the season, the tick season. But we also speculated that, okay, now they're attacking all these other species as well, right? So what if we applied at a different season where we know the ticks are still on the landscape, might not be active, but they're on the landscape. There's also another active stage that we could be attacking simultaneously. And then there's a lot of other options for spraying. So like if you see in this picture, this is one of our spray sites for the fall. All the foliage is pretty much down, which makes it really accessible to access that leaf litter with something like a high pressure spray rig. As you can see in this picture, my wonderful technician, Jamie Cantoni, is doing an excellent job at spraying that 1 meter zone. And she can penetrate right down to the leaf litter, which is where the adults and the nymphs are hiding out. And then at this time too, yes, there are some pollinators that will be out. That being said, it's not the same activity level that we see in the spring. So a lot of those pollinators, non-target species, the monarchs and things like that, their crazy active period is in the spring. So this kind of subdues a lot of that impact by removing that questing activity or the activity period of the non-target species, and even some of the ones that migrate out of the state at that point. And so we're focusing, like I said, on the dormant nymphal stages and then also the active adult stages at that time. For this particular project, we were looking at the application of Demand, which is a lambda cyhalothrin-based synthetic pyrethroid. And we did it in let's see, 2 different substrates and 2 different applicators. So we did a high pressure spray rig and a backpack blower. And then we did it in liquid formulation, which was a concentrate diluted out. And then we also did it with a granular formulation. So this was not only, we wanted to see the differences in seasonal application, but we also wanted to see the difference in the solution as well as the applicator. Excuse me, we did a standard spring application, so when it's traditionally held. We did a fall application post-frost when foliage was down. And then we also did a combination to see if like maybe there'd be a greater effect there. And we did it across 70 properties total. And I will also say, anecdotally speaking, these projects people are really excited about. So typically when we request homeowners to participate and we need 70, like a recent project we did, we need 70 and we got 300 people who wanted to participate. So it's really reassuring to see that people they want to treat their backyards and they want to reduce their health risks, but they really also want to see a more sustainable practice in place. So this is the distribution of the type and application, and then the seasonal application as well. And I should also note that this was a pilot study to determine if we could actually see any impacts in the fall application versus spring, and if they would be significant enough to pursue further. So right out of the gate, nothing surprising here. There's a reason why we spray in the spring, because it's highly effective at reducing questing nymph densities. So with these applications, we collected no nymphal ticks. However, you can see with our water application or control that the typical trajectory of questing nymphal activity was normal. So this was highly effective for across all 3 different types of high pressure, low pressure, backpack, and granular. As for the fall only spray, you'll see that it is actually very similar. So we had the application of the spray, and it seems like the only one that showed activity or questing behavior or collection of nymphs was in our control properties. I will say that there was a couple nymphs that were collected in our granular only application for the fall. So once again, a difference in the substrate itself, not necessarily in the application, but for all of our other treatments using the liquid formulation, no nymphal ticks were collected. And I should say too, so we sprayed and then we sampled the following spring. So this isn't to say that we were collecting questing nymphs in the fall because they're not active. We collected the following spring to see how it would impact them. And then finally, when we did our fall and spring application, once again, had very similar impacts. We saw a reduction or no nymphal ticks collected at either of those sites, and only one was collected for the granular treatment. From this, we determined a couple things. The very first and basic thing was that when we're looking at things like high pressure versus backpack sprayer, they both had multi-season impacts, regardless of whether we applied them in the spring or the fall or a combination thereof. And that was for the liquid formulation. I will say with these two, before I get too far ahead of myself, this was the pilot study, so we really wanted to see that if it had an impact on questing nymphal densities, but we did not measure pollinators. We're actually doing that right now with the secondary study. So if you're interested in that, I can talk about that afterwards. High pressure was definitely the most effective, and for a couple reasons. So not only if you know anything about high pressure versus backpack or low pressure spraying, so the low pressure spraying is really more of a fine mist. And so when you have, especially in the spring, when you have dense foliage, it's really hard for it to kind of pervade through that layer and get down to the leaf litter where we know ticks are hiding out. It might be more effective at something that's a little bit more airborne, so like mosquitoes and things like that. But for ticks, we really need to penetrate that leaf litter area and also the thick, dense understory that typically they hide out in. High pressure spray rig just does a better job at that. And especially when we're trying to get 1 meter in or 3 meters into the woods, we need that distance covered as well. It's just overall, the high pressure spray rig was highly effective. So not only because it breached the hospitable areas, but also got underneath the leaf litter, which for this to work effectively on the nymphal densities that we see in the fall, it needed to penetrate that because that's where they're all quiescing. So in conclusion of that study, and like I said, it was a single year, well, I guess it was a year and a half to get all the seasons covered, kind of pilot study, we determined that the demand CS or the lambda-cyhalothrin was highly effective with high pressure delivery. However, all 3 of them worked really effectively. The granular was also effective, but you just saw the numbers rebounding a little bit at the end. And that might just purely be because it's not as pervasive. Obviously, it's a granular formulation versus a liquid formulation that can have a more substantial coating on the surface area versus granular. It's more contact driven. So that might also be something that like maybe you have to increase the amount that you put on the landscape for it to be more effective and have greater coverage. But overall, the synthetic pyrethroids in this particular case were effective across all the seasons, not just specifically this traditional spring application. And so this is really significant because not only are we telling people that they can choose a different season and it could be just as effective, but you're also telling them that they don't need to be out actively questing in order for them to make contact with this product and to reduce those tick densities in that paradomestic habitat. So we can apply it in the fall and we can see a significant reduction in those nymphal stages. That being said, this is also a single-year study. We know that adults are out questing during that time as well. So not only we're just attacking the dormant nymphs that are in the leaf litter, but we're also getting any questing adults as a result too. And so these longer term studies might be able to better quantify that. If you're attacking two different stages contemporaneously for a tick species that has a 2-year life cycle, you can see longer term impacts. And then on top of it, it's really important that we note that this is another option for people who want to treat their backyards and reduce their public health risk, but they don't want to have that sustained impact on non-target species or beneficial species. I know from myself personally, I have a lot of people who are very concerned about their bees in their backyard. And so knowing that they can maybe treat at a different time frame where there's not going to be as significant of an impact is really important to them. Same thing with, like I said, other non-target beneficial species like monarchs. And so we have a lot of other initiatives in place to help sustain their habitats and their structures and things like that. So this might be another way for us to do this effectively and sustainably over time, while also reducing the public health risk of ticks in people's backyards. And I will also say, I've been include a slide about this. So all the studies that we're doing are looking at individual applications and treatment strategies and things like that. And the idea is that we do these initial studies to see if they work or if they're effective or if they need to be optimized. And then the next step is to kind of maybe test that optimization. And then furthermore, then we start to integrate them. And so with these particular projects, they were of significant interest because they are something that directly would benefit homeowners, communities, and neighborhoods. So we know people are treating their backyard, and we know that they hire pesticide applicators to come in and do what they think is best for not only the homeowner, but for their application process. But if we can make that a little bit better, still reducing that public health risk for homeowners, but also reducing the environmental impacts as well, that's pretty significant. And especially when we look at some of these landscape-wide or ecologically based approaches, we know more now than we did before. So we should be able to optimize, advance, and adapt these strategies and work with not only homeowners, but pesticide applicators as well to make better strategies for reducing this paradigmatic risk. So I know I went through that very quickly, but I also want to leave plenty of time to talk to everybody about other questions and stuff like that. But I also want to give a shout out to this amazing tick team. And we also have a suite of seasonals who have helped us over the years as well. And I couldn't cram them all into here, but we have several people both still here and retired that have helped on these projects. And so I had to give them a shout out. Thank you all so much for coming and listening, and I can take questions.
Thank you so much, Megan. That was a great talk. If anyone has questions, yeah, feel free to ask them or drop them in the chat, raise your hand, whatever works for you.
Great talk, Megan. I have a question, I guess, about the process. So looking at sampling and spraying in people's backyards, how was your IRB process in that? Was there a lot of oversight? Did you need like a lot of detailed approval? We're kind of working through that now. And so any insight that you have would be wonderful.
Yes, so I will say we do not have an IRB in place at the Ag Station and did not need it for any of these particular projects, mostly because we do not dabble in the medical aspect or the physical disease manifestation aspect. And so therefore we don't handle anybody's private health information. So all the work that we do is purely contacting homeowners to see if they would like to participate in the project. And then that's pretty much it. So it's just having access to their properties and they're participating in it.
Do you ask them any questions in particular, like throughout, other than like to recruit?
So we've had a couple different strategies for recruitment, but honestly, our most effective strategy is posting it on town Facebook groups.
Okay.
Or reaching out to their public health departments. And so, like I said, our most recent study, which is actually a build off of this particular one, we needed 75 homeowners and we put out a Facebook post and then their public health department posted it. And we ended up with over 300 people wanting to participate. So it worked out really well. And I think having the public health department backing was really important because it was a trusted institution in their town versus if they didn't know about us in particular, they might have been a little bit.
And that makes complete sense. Thank you.
Yeah, no worries.
So Megan, really nice work. I'm curious if you have plans for a follow-up longer term study or if that's.
We are in the thick of it right now.
Oh, good. Okay.
Yeah, so we're actually in year 2 of a follow-up study to the seasonal spray application piece of it. So looking a little bit more at that longer term impact. And then also we're doing more pollinator surveys too, to see if we can address the impact to those directly. Because obviously that was a question. The first question was, will this have any impact on tick densities if we spray at a different time? And now that we know it does, we want to make sure that it doesn't impact pollinators by spraying in a different season. So yeah, we have, I believe, 1 more year left on that, and then we'll have all that data up and ready for you guys.
Oh, fantastic.
Does anybody have any more questions? I'll give it another minute or so.
Yeah, I have a question, Megan. Thanks for that presentation. Just regarding your follow-up, I was just wondering, when you're looking at impacts on pollinators, are you doing like pollinator observations, or how are you trying to measure that impact?
So I will also preface this by saying I am not the pollinator expert here at the Ag Station, so I deferred to a couple other members of our team who are much more proficient. And they recommended that we do blue vein trapping, which are basically like patches that you put in the shrub layer of these ecotonal areas to do a measurement of how many different species or densities there are. And so we're doing it more as a comparison across treatment types. So we're placing them out for 48 hours, collecting them, and then sorting and identifying the different species and doing a comparison that way.
Cool. So you can look at abundance, but you also can look at the diversity, which is pretty cool too.
Exactly. So we're trying to time it with the spray application. We're doing a pre and post measure, and then just across the treatment types as well. And then doing it in both the spring and the fall to see, because obviously there are still non-target and beneficial species that are out in the fall as well. But if we can do a comparison across those 2 seasons as well, that would be beneficial too.
Another question I had was, have you ever measured the residual activity of the pesticide? Like, have you put out filter paper or some way to look at the analytical chemistry of what remains in the environment and for how long?
Yes, you guys are asking all the questions that we're doing right now, so this is perfect.
Okay.
So we actually, after we did the spray application in the fall, because it's like, the physical breakdown is going to be very different in May and June than it is going to be in November, December, January. And so we did leaf litter samples at all of our fall applied, chemical applied spaces, leaf litter and soil samples. And then we brought them back to our analytical lab, and they are processing them for the AI as well as the metabolites from them as they break down. And then we're going to go out, I think in, my calendar says 2 weeks, and then collect from the same sites, leaf and soil samples, and do the same testing to see, like I said, not only what the presence and concentration of the AI is, but also if any of the metabolites are present, to kind of answer that question of how it's breaking down over time, but also is the efficacy truly from the fall application, or is it from the residual impacts that are still on the landscape come spring? So yes, we will hopefully be able to answer that question.
Yeah, great. Looking forward to hearing what that looks like.
Okay, well, if there are no more questions, I guess everyone has Megan's contact information, so you can reach out to her another time if you come up with anything. Thank you so much, Megan. That was excellent. And we will see all of you on April 9th for the next Work in Progress meeting.
Thanks guys for coming. I appreciate it.